Imagine a vast library filled with countless books, each containing a unique story. Our genetic blueprint, DNA, is akin to this grand library, holding the instructions for life. PCR, or polymerase chain reaction, is a molecular technique used to amplify a specific DNA sequence, much like photocopying a single page from a massive library. Primers are the key to this process. They are short, single-stranded DNA sequences that act as guides. They direct the DNA polymerase, the enzyme responsible for copying DNA, to the specific region we want to amplify. Think of primers as bookmarks, flagging the starting and ending points of the DNA segment we're interested in. The design of these primers is crucial. A well-designed primer ensures that only the desired DNA sequence is amplified. The length of a primer is a critical factor that influences its performance. Primers that are too short lack specificity. They may bind to multiple sites on the DNA template, leading to non-specific amplification. Conversely, primers that are too long can be difficult to synthesize. They may also fold back on themselves, forming secondary structures that hinder their binding to the template DNA. The ideal primer length typically falls between 18 and 25 nucleotides. This range strikes a balance between specificity and ease of synthesis. A useful analogy for understanding primer length is to think of a key and a lock. A key that is too short will not engage all the tumblers in the lock and therefore will not open the door. Temperature plays a crucial role in PCR, particularly during the annealing and melting steps. Annealing is the process where primers bind to their complementary sequences on the template DNA. Melting, on the other hand, refers to the separation of the double-stranded DNA into single strands. The annealing temperature, TEM, is the temperature at which half of the primers are bound to their target sequence. Selecting the appropriate annealing temperature is crucial for successful PCR. If the annealing temperature is too high, the primers may not bind to the template DNA at all. If the temperature is too low, the primers may bind non-specifically to other regions of the DNA, leading to amplification of unwanted products. Think of annealing and melting as a delicate dance between the primers and the template DNA. Finding the optimal temperature ensures that the primers and the template DNA move in harmony, leading to successful amplification of the target sequence. Section 4. GC content. The Goldilocks principle in primer design. GC content, which refers to the percentage of guanine, G, and cytosine, C, bases in a DNA sequence, is another important factor to consider in primer design. Guanine and cytosine form 3 hydrogen bonds between them, while adenine, A, and thymine, T, form only 2. This means that DNA sequences with higher GC content are more stable and have higher melting temperatures. The ideal GC content for primers typically falls between 40% and 60%. The Goldilocks principle applies perfectly here. The GC content should be just right. Maintaining the GC content within the optimal range ensures that the primers bind to the target sequence with the right amount of affinity. Section 5. Beware the hairpin, navigating secondary structures. Secondary structures, such as hairpins and dimers, can significantly impact the performance of primers. Hairpins occur when a single primer molecule folds back on itself, forming a loop-like structure stabilized by base pairing between complementary nucleotides within the primer sequence. Dimers, on the other hand, form when 2 primer molecules, either identical or different, bind to each other due to complementary base pairing. These secondary structures are undesirable because they compete with the binding of the primers to the template DNA. To avoid the formation of secondary structures, it is crucial to carefully analyze the primer sequences during the design phase. Section 6. Troubleshooting your PCR when primers misbehave. Even with careful design, PCR reactions may sometimes not go as planned. Troubleshooting a PCR experiment often involves identifying and addressing problems related to the primers. One common issue is the absence of amplification, indicated by the lack of a product band on an electrophoresis gel. This could be due to several factors, such as incorrect primer design, degradation of primers, or errors in the PCR protocol. Another common problem is non-specific amplification, where multiple bands are observed on the gel instead of a single distinct band. This indicates that the primers are binding to multiple sites on the DNA template, amplifying unintended regions. To troubleshoot these issues, It is crucial to carefully review the primer design, ensure the quality and integrity of the primers, and optimize the PCR conditions. Experimentation is key in PCR optimization.